So this brings us to the very last talk of this year's CGC meeting, and I'm sure you will agree with me when I say that this was another fantastic CGC meeting. It's sad that it's coming to an end, but I'm absolutely committed to come back again next year, and I already look forward to it. So you heard about many different interesting projects and initiatives that you could be participating within CGC. But I will be telling you today about an initiative that I'm involved with within the clinical genome resource, ClinGen, and something that can also be of interest, especially to those of you who are working with pediatric tumor variants, and I especially recommend our trainees to consider being involved. So I will be talking about curation of variants associated with pediatric tumors within the clinical genome resource. So we are familiar with wonderful contributions that ClinGen has been making over the years to developing resources to clinical genetic and genomic testing interpretation through multiple projects within ClinGen related to gene disease validity curation, curation of pathogenicity, clinical actionability, dosage sensitivity. But what we are mostly familiar with are different initiatives within ClinGen that are related to constitutional genetic testing and hereditary genetic diseases. So it's a little less known that there is also a somatic initiative within ClinGen, and there is somatic variant curation work group that has been very active and was started in 2015. It is led by Shashi Kulkarni and Subha Madhavan. We have more than 60 members from different academic institutions, from industry, and we have a wonderful coordinator in Shruti Rao from Georgetown University. So here are some of the accomplishments and important milestones from Somatic Work Group. In 2017, the group developed and published what we called MVLD standard. It stands for Minimal Variant Level Data Standard, and I will explain what it is in the next slide. We also published review describing activities and initiatives within working group. Very importantly, we established collaboration in CIVIC. So we just had a really nice presentation about CIVIC and curation of variants in CIVIC. We decided early on that we wanted to select CIVIC as a curation platform. So we have a really productive, mutually beneficial collaboration with CIVIC. We are organized into curation task forces, focusing on different tumor types, actively recruiting curators, conducting curation training, adding evidence into CIVIC. Code has been developed to transfer CIVIC variants into ClinVar. Again, another thing that was just mentioned by Killian in her really nice talk. And we are developing protocols for somatic gene and variant curation. So I mentioned that one of the early initiatives within Somatic Work Group was development of minimum variant level data standard. So we proposed that minimum data elements and standardized formats that we should be collecting and gathering in knowledge bases and our shared resources when it comes to somatic variants. And this consists of data elements that describe the variant itself and also its significance, clinical significance for a particular tumor type. And this was published before ASCO CAP guidelines were published for somatic variants, and after the guidelines were published, we coordinated MVLD definitions to incorporate the ASCO CAP guideline principles. This is just a screenshot from CIVIC showing that ClinGen is one of the organizations within CIVIC. And then for the the rest of my talk, I wanted to focus on Pediatric Somatic Taskforce because this is a task force where I'm actively involved within Somatic Work Group. So we decided that one of the focus areas in Somatic Work Group will be pediatric tumors. And that's because they are completely different from tumors that you have in adults. And there are different mutations in pediatric tumors in completely different genes. And they are currently very sparsely curated in CIVIC and in other cancer knowledge bases. So we decided to focus on this void and this gap and to address that within a separate task force in Somatic Work Group. So our vision is to engage oncologists, molecular pathologists, PhD laboratory geneticists, curators who are a lot of our trainees, to oversee, coordinate, and curate childhood cancer associated variants so that we can develop clinical grade interpretation knowledge base. And some of the immediate tasks and goals that we focused on was to identify and prioritize specific pediatric cancer types, genes, and variants for curation based on their incidence and clinical significance. And also we work on curation and to provide consensus assertions regarding associations of specific genes and variants with diagnosis, prognosis, and drug response. So in a previous talk, Helen very nicely defined what the assertions are. So we are working within our task force on creating those assertions for variants that we prioritize for curation. This is just a composition of the pediatric task force. So I lead this effort together with Anshumoy Roy from Baylor College, from Texas Children's Hospital. We have 10 different expert members from different institutions. As I mentioned, we work very closely with bioinformatics team in Civic, and then Deb Ritter is a bioinformatician with ClinGen who works with us. We have 8 curators, and coordinators is Shruti Rao. And then we also work very closely with human disease ontology to ensure that the best classification systems are being used, if tumor classification system is being used when we curate these variants. One of the early tasks was to prioritize the genes and variants for curation. So we prioritized so far 44 genes and variants from 23 different tumor types for curation. And then this is our working model. We try to ask curators to select a tumor type or variant or gene that they're particularly interested in, and we try to pair our curators with expert members who then help to guide curators in their curation efforts. Expert member will help curators to select publications for curation, will review their work, and that way curators are able to learn from the expert member and be more guided in their work. And we have monthly conference calls, which are working calls. So during those calls, we discuss specific variants, pre-selected variants and during the call, we make a point to make final assertion regarding that variant. So evidence and information is gathered ahead of time. The evidence items are entered in Civic. Short summary statements are pre-entered in Civic. And then everything gets discussed during the call by the entire task force membership, and the assertion is finalized and entered in Civic. So I want to stress this because I find these conference calls and these discussions about different genes and variants very educational to all the members of the task force, because we have a variety of expertise within the group. This is just a screenshot from Civic showing our members and some of their activity, and the quick summary of the number of evidence items that have been created so far by task force members and number of assertions that we have created so far. And I mentioned earlier that we work very closely with human disease ontology. So this is an ontology system that was selected by Civic for tumor classification. So we work with this group to ensure that there is appropriate classification available for pediatric tumors. And for now, we selected ACDO as a classification system that we would like to use, and when we curate our variants. So we worked with HDO to make sure that it becomes available in HDO and therefore also available in Civic. One important recent development within ClinGen was that FDA recognized ClinVar as a genomic database, and also in the process, it recognized ClinGen insertions which are made regarding variant pathogenicity by ClinGen variant curation expert panels. That prompted organization of a large number of variant curation expert panels within ClinGen that are focusing on different disease areas and different genes, different diseases. So we decided within Pediatric Task Force that we will also start organizing into expert panels that will be focusing on specific genes or specific groups of variants. So we are now in the process of generating first expert panel within the somatic domain in ClinGen. And this is just to illustrate that there is a very structured, very organized process of generating an expert panel, and you have to go through a process where you define your scope and your membership. You define your curation protocols and curation rules. You pilot and test your rules on a list of test variants, and you summarize all that in an application that has to be approved within ClinGen, within appropriate bodies in ClinGen. So we are working on adapting this VCEP process for somatic domain and for our first expert panel within Pediatric Task Force. And in terms of the scope, we decided to first focus on NTRK fusions, and there were a couple of really excellent talks during this meeting about NTRK fusions, so there is no need to introduce these variants. Just to say that these are fusions that act as very potent oncogenic drivers, and they're present in a variety of different tumor types. So the reason for selecting NTRK fusions was primarily their high clinical significance. We know that there are FDA-approved inhibitors with remarkable anti-tumor activity targeting tumors that have NTRK fusions. And then although these fusions are generally rare, they're very common in certain pediatric tumors, including infantile congenital fibrosarcoma, pediatric high-grade glioma, and some others. So the scope of this expert panel will be curation of all known NTRK fusions, and clearly it will be a complex task because there is a large number of known individual NTRK fusions. And in addition, they are found in a large variety of different tumor types, both in pediatric and adult population. So within scope of the expert panel will probably be all different NTRK fusions in all the variety of different tumors where they occur. And in addition, there are also resistance mutations that have been described after treatment with inhibitors in these NTRK fusions. So we are probably going to include that into the scope of our expert panel as well. So we are early in the process, but we have started defining curation data and curation elements and curation protocols for NTRK fusions. And so that is one of the new ongoing initiatives within the Pediatric Task Force. So just to summarize, what has Pediatric Task Force done so far, and where are we going? We currently have more than 20 members from more than 10 different institutions. And we prioritize 44 genes and variants for curation. We entered more than 140 evidence items and 5 assertions in CIVIC. And we have our curators working with expert members on genes and variants of interest. So we get feedback from our curators that it's a really nice collaboration with a senior mentor and a wonderful way to learn. We make new assertions during each monthly call, so those are working calls that are also very educational. We are working on providing appropriate tumor classifications for pediatric tumors through HDO within CIVIC. And then we are in the process of forming the first somatic domain variant curation expert panel, which will be focusing on NTRK fusions. So if this is something that might be of interest to you, if you work on pediatric tumors, we are always looking for new curators, or you are welcome to just join one of our conference calls that are completely open and to hear a little more about what we do. So please get in touch if you might be interested. And thank you for the opportunity to tell you about this initiative.